Thank you very much for your introduction. Today I would like to introduce about some methane production by microbial ability, named as direct interspecies electron transfer. So methanogenesis is a microbial reactions that can convert complex organics into methane. So when all of the electron acceptors are depleted, except for CO2. These reactions can be used for industrial purpose or waste minimization, and also this reaction is very important in carbon conversion and global warming. Today, I would like to focus on waste minimizations based on methanization or methanogenesis. So methanogenesis actually can occur by syntrophic association with many bacteria or archaea in natural or in engineering systems. The most important reactions actually is done by methanogenic archaea. They have ability to convert CO2 into methane using electron carriers. However, methanogenic archaea cannot work very well without help of other types of bacteria called as fermenting bacteria. So basically, methane formation is a kind of syntropy between fermenting bacteria and methanogenic archaea. So this reaction can be explained as some electron transfer. Basically, electrons deposited in complex organics can be transferred to the methanogenic archaea using diffusive electron carriers, such as hydrogen or fomate. This is a very important reaction for forming CO2 to methane. However, this reaction is mostly dependent on the diffusion, mass transfer diffusion of electron carriers. However, thermodynamically, it is very difficult to achieve high concentrations of hydrogen or fomate because the formation of hydrogen or fomate can be possible when these two product concentrations are very, very low. So principally, it is very difficult to achieve high rate of methane production. So, however, these reactions, the diffusion of electron carrier, instead, electrons can be directly transferred from the fermenting organism or oxidizing bacteria to methanogenic archaea. We call this reaction is direct interspecies electron transfer. These reactions can be manageably used for methane productions. So one thing I can stress is the transfer of electrons directly without mediating a diffusive electron carrier. This one is more efficient because one reason is the bacteria cannot produce some of enzymes that involved in the hydrogen or fomate production, and also electrical conductance is more efficient than molecular diffusion of these molecules. There are three main possible ways for direct interspecies electron transfer available. The first one is using conductive pili. This pili is a kind of electron wire that can directly transfer electrons from one bacteria to other microorganisms. Second types of electron transfer using electron transfer proteins located on the surface of the bacteria. And the third type is using some material that can conduct electrons, such as granular activity carbon or biochar or carbon plus and so on. So I would like to introduce a third part of the electron transfer for methane production. So granular activated carbon traditionally are used to enhance methane production because this material can absorb some of toxic chemicals that can inhibit methane production. So it is a good way to increase methane production. Another characteristics of the granular activated carbon, this material has a very high specific surface area that can attach some of the microorganisms that can facilitate methane production. And also, granular activated carbon is a good conductor of electricity. And these days, some of the researchers reported this material can enhance direct interspecies electron transfer for methane production. However, most of the studies conducted in just batch reactors based on pure culture strains. So it is not certain whether this theory can be realized in real systems, real environmental engineering system, in which many kinds of microorganisms are present in the feed and also in the reactors. And also there are some of indigenous microorganisms in the reactors. So we are not sure whether the addition of this can facilitate some of enrichment of some microorganisms are involved in direct interspecies electron transfer. So there are some research questions associated with this. The first one, I would like to answer is, we are not sure whether just addition of the GAC in the continuous flow reactors can facilitate diet or not. And the second one is, the bacteria or archaea involved in the diet can be enriched in the reactors. And the third one is, what are some of the some benefits in engineering perspective or social perspective? To answer those questions, we operated two reactors, actually continuous flow reactor. One is just a normal anaerobic reactors without GAC, granular activated carbon, and the other one is the flow reactor, continuous flow reactor with GAC. We added 1% of the GAC, weight per volume, to the reactors. And also we use the acetate as the sole carbon for the microbial reactions. So first we trace the performance of the reactors, such as methane production or COD reductions. And we evaluate the relative contributions of methane production between suspended biomass and attached biomass. And also we traced microbial communities for the bacterial populations and the archaeal populations. So lastly, we used the bioelectrochemical analysis for finding some of direct evidence of direct interspecies electron transfer in the reactor. This is the profiles of methane production rate as a function of reactor operation. As you can see in here, the red colors indicate a methane production rate for the reactors with GAC, and the blue line indicate for methane production rate for the reactors without GAC. So as you can see that the reactor with GAC showed much higher methane production rate. The rate is equivalent around 1.8 times as compared with just a normal bioreactor. Secondly, we traced the COD analysis. I found that the reactor with GAC demonstrate a much lower COD level in the effluent. The second question is about what is the main player for higher production of or higher methane production rate. For this analysis, we took biomass samples after end of the reactor operations, and we used just a serum water with the GAC and the bulk solution. And the second one is just bulk solution, and third one is a GAC. So left figure shows the cumulative methane production as a function of time. As you can see that GAC plus bulk solution show the highest cumulative methane production, second bulk solution, third the GAC. However, if we normalize by the biomass concentrations for this data, we can see that the GAC show the highest specific methane production rate. This data indicate that the higher methane production rate mostly due to the biomass attached to the GAC. Secondly, we traced the microbial communities. We found some of the specific enrichment of the microorganisms. We found mostly Geobacter and Thora and Godunia. However, Thora and Godunia, I thought they are not the active members of the direct interspecies electron transport. So, however, Geobacter are commonly found in microbial fuel cells, and many researchers demonstrate that the organism can transfer electrons extracellularly to the electrodes. So we deduced that Geobacter mainly enriched in the attached biomass, and they are involved in the transport electrons to the GAC. And we also traced the methanogenic archaea. We found the specific enrichment of the methanospirillum, but we are not quite sure whether these organisms have an ability to can accept electrons from the GAC to make methane. So to verify these possibilities, we did a network analysis. We can see that left one shows just a control reactor, means reactor without GAC, and the right one for the network analysis for the reactors with GAC. As you can see, without GAC, the two microorganisms, Methanospirillum and Geobacter, actually does not correlate significantly, and also located in different modules. However, the reactor with GAC, so as you can see, these two microorganisms are within the same modules and have some of significant correlations. This data demonstrate these two microorganisms are really important and correlated for higher methane productions. So lastly, we would like to get some of direct evidence for diet in the system. We set up 3 electrode cell systems by using the biomass attached on the GAC, and we inoculated the biomass in this bioelectrochemical cells, and we incubated for 3 days by addition of acetate. So as you can see, by increasing potential, we can find the specific current generation at these 2 voltage range. So specifically, we found some of some oxidation peak at minus 0.36 volt. Actually, this voltage corresponds to the oxidation of acetate into the CO2, and also we found some of reduction peak at minus 0.32 volt. This peak corresponds to the CO2 reduction into the methane. After removing the biomass, we also observed the voltammographs. However, in this case, no oxidation peaks and no reduction peaks were found. So this data strongly suggests that the reactions is occurring biologically, and also this reaction shows some of the direct information for methane production by electron transfers. So now I want to conclude our research. So first, I want to say that supplementation of the GAC can increase the methane productions up to around 2 times. And also, by addition of the GAC can enrich some of the specific microorganisms involved in the diet, for example, geobacter or methanospirillum. And also, cyclic voltammetry is a good tool for demonstrating the evidence of the diet in the systems. So finally, I would like to thank some of the researchers in our groups, Jung-Moon Park, Jung-Ryul Kim, and Hyun-Jin Kang, and Sang Lee. Thank you for your listening.
Thank you very much, Mr. Park.